So, the floor is yours.
Thank you. Well, the presentation.
It will come, I'm
Yeah.
sure. Andrea, where are you from?
From Spain.
From Spain.
Yeah,
All right.
yeah. But I'm working in Australia with Leszek, Children's Medical Research Institute.
All right.
We are working on the development of a gene therapy for CTNNB1 syndrome. So today I will try to introduce a little bit about gene therapy so everyone can be on the same page. And then I'll show you some of the results that we obtained in the lab. First of all, I'd like to introduce about genetic disorders. They impact more people than HIV and cancer combined. And this is quite surprising because normally people don't know much about genetic diseases. So actually rare diseases or rare genetic diseases, although they are rare alone, when they are combined, they are not rare anymore. Indeed, there are over 7,000 known genetic diseases, and more than 8 million children are diagnosed worldwide each year. And many of them cannot reach the 5th birthday. So this is a huge problem. However, we are in an exciting time where we have been witnesses of a big revolution in genetics since the discovery of the DNA structure. Now we are able to modify the DNA. We can copy paste some parts of the DNA in other parts. We are even able to read the human genome. There was a project called the Human Genome Project that wanted to read the whole DNA in the human. Of the humans, and it costed millions of dollars, and it took over a decade to do that, and now we can do it just in a couple of days, and it is way cheaper. So we have progressed a lot in these technologies, so we are in a very good time. However, although we know a lot about many of the genetic diseases, we have the tools to diagnose them. There is a big gap in which many of those genetic diseases, as you know, cannot be treated nowadays. And here's where gene therapy could help solve this gap. And what is gene therapy? Well, gene therapy, the simplest way to describe it is just the use of genes as medicine. So we basically, instead of using drugs, we use a piece of DNA that will be used as a therapeutic, and we can package them in what we call a vector, which will be like a delivery box that contains the address where the therapeutic should go in the body. And that's why we try to do with gene therapy. So instead of needing surgical tools, a surgeon and new organs, many times with a vector and a therapeutic gene, we could solve or treat a disease. And this is, there are already some examples of this in the clinic. These three therapeutics are treating different genetic disorders for the eye, for the spinal muscular atrophy, and also for hemophilia B. And the three of them use the same vector, which is the adeno-associated viral vector, or AAV. And we use, we also are using this vector because it doesn't cause any disease in humans in the nature, and you can easily take out all the viral genes that it contains, and you can insert in there the therapeutic genes that you want to use. So this is another representation of the same thing. We can remove what's inside of the AAV virus and convert it turn it into a vector that will be able to insert the therapeutic gene in the target cells. So I'm gonna explain a little bit about DNA. Well, you all know, obviously, CTNNB1 is the gene for beta-catenin protein, and the DNA is like the set of instructions that we all have in the cells to be able to build all the proteins that we have in the body. So there is a special chapter that contains the information to build the beta-catenin protein. And this chapter is the CTNNB1 gene. And a single typo in this chapter could make the beta-catenin protein not to be produced properly. And that is what could lead to disease, because the functions of the beta-catenin protein will be dysregulated. And beta-catenin is involved in the communication between cells and also in the communication between cells and the muscles. And that's why many symptoms are associated with cognitive and locomotor defects. So with gene therapy, we can put again the chapter that has a typo corrected inside the box, inside the vector, and reach the cells that are affected, and then those cells will be able to produce again the beta-catenin protein properly. And we can also use some regulatory elements that here they are represented as tools that will allow a faster or more robust production of the protein. So when we designed the AAV-mediated gene therapy for CTNNB1 syndrome, we removed all the viral genes from the AAV vector, and we introduced the therapeutic gene, which is constituted of the set of the instruction of the CTNNB1 gene without the mutation, and some regulatory elements that could increase the production of the beta-catenin protein where we want it to be, and also avoid the expression in other tissues where we don't want to overexpress beta-catenin. Two organs that we don't want beta-catenin to be overexpressed in are the liver and the dorsal root ganglion, and this is because it has been observed that the overexpression in these tissues could lead to toxicity. So we wanted to test if we could use these regulatory elements to avoid the expression of beta-catenin in these organs. So you can see the green is the production of the protein. So the more green you see is the more protein that is produced. And in the liver, we tested 2 different doses of vector, a low and a high dose. And in the dorsal root ganglion, which are the sensory neurons that are at both sides of the spinal cord, we used the highest one. And when we included these regulatory elements, we could avoid the expression of the beta-catenin. So we included these regulatory elements in all the vectors that we designed. So we designed a set of different therapeutic constructs that will include the healthy version of the gene and different regulatory elements, because not all the regulatory elements fit in an AAV vector. So we had to do different combinations to get the best one. And then we tested them in patient-derived neural models. These patient-derived neural models were generated from blood of a patient. These blood cells were reset. It means they were not blood cells anymore. They were pluripotent cells that could be turned into any cell type. We could turn them into muscle cells, into lung cells, but we wanted them to be neuronal cells. We wanted them to be neuronal cells because we want to know what's happening in the brain of the patients. And we wanted to use patient-derived cells because these cells will have the exact same mutation as the patient. So these neurons that we generate from these cells will act similarly as they would act in the patient. So from these pluripotent cells, we could generate 2-dimensional neurons, but also 3-dimensional brain organoids that are like mini brains that we can maintain in the lab, and we can test the therapeutics there. So we generated 2 different models, neuroprogenitor cells and brain organoids. we tested firstly the neuroprogenitor cells, and we wanted to check first that the neuroprogenitor cells were acting similarly to what we would expect from a patient, which is a downregulation in the beta-catenin production. So on the left-hand side, you can see a decrease in the expression of CTNNB1 in these patient-derived neuroprogenitor cells compared to the wild-type control cells that don't have the mutation. And we could also see, I don't know if you are able to see it, but we can also see under the microscope some differences between the wild type and the patient-derived neuroprogenitor cells. So a neuroprogenitor cell is supposed to have some projections because it is starting to differentiate into a neuron, and this is not observed in the patient-derived neuroprogenitor cells. So we are already starting to see some phenotypes that that could be saying us that this is a good model for CTNNB1 syndrome. And then we also observed the dysregulation of a protein that is in close interaction with beta-catenin, which is the encadherin. So we checked that we verified that this model could be used to test the vectors. So again, we differentiated another set of cells, and we treated them with the vectors that contain the different constructs. And we preselected 3 of the AAV vectors with the 3 different constructs because they were able to lead to a higher increase of the beta-catenin protein, well, at the level of messenger RNA, actually. So these are AAV construct 2, 4, and 6. So we repeated another experiment with only these 3 constructs, and we observed this increase in the beta-catenin expression at the level of messenger RNA compared to both the untreated patient-derived neuroprogenitor cells and the wild types. And we also checked for the function of beta-catenin. So we checked for the expression of DKK1 and MYC, which are 2 target genes of beta-catenin when it goes into the nucleus, and we could see a partial increase of the levels of these target genes. So we could see that there was also a bit of improvement in the beta-catenin function. After that, we move into brain organoids because it will constitute a better model because it contains not only neuroprogenitor cells, but also other cells that are present in the human brain. And again, we wanted to check that this will constitute a good model for CTNNB1 syndrome. So we also checked the expression of beta-catenin at the level of the messenger RNA. On the left-hand side, you can see again the drop in the expression in patient-derived brain organoids compared to the controls. And we also check it at the level of protein. So again, the more green you see is the more protein. And you can see that there is a significant drop in patient-derived brain organoids compared to wild type. And we also check for the expression of 2 other proteins that will indicate for the function of beta-catenin, synaptophysin, which is a marker for synaptic activity, so the communication between neurons, which was also downregulated in patient-derived brain organoids, and GFAP, which is an indicator, a marker for cells. And with this indicator, well, with this marker, we could check the activity of beta-catenin in proliferation of specific cells. And again, this was also downregulated in patient-derived organoids. So after that, we again treated the brain organoids with the different AAVs. And in this case, only the AAV that contained the construct number 4 was able to significantly increase the levels of beta-catenin messenger RNA compared to the untreated patient-derived brain organoids, and these levels were comparable to the wild type. And we also checked it at the level of protein expression, And again, you can see the difference between the wild type and the patient-derived brain organoids, and then how the recovery when you treat the brain organoids with the AAV containing the construct number 4. So with all this information, we selected the AAV with the construct number 4 to move forward into studies with animals, but Duško will talk about it later, so I'm not gonna talk about it now. But we also did a preliminary safety assay in a wild-type mouse. This wild-type mouse doesn't have the mutation in beta-catenin protein, in the beta-catenin gene. So we wanted to check if the overproduction of beta-catenin due to the inoculation of the vectors will lead to toxicity. So we inoculated the mice using the same route of administration that we want for the clinic, the ICV administration, with 2 different doses, a high dose and a super high dose. The high dose is the highest dose that was used for the efficacy study that Duško will talk about. And super high dose is like double that dose. So we inoculated the animals and monitored them for 8 months. Weekly, we checked for the behavior, the physical wellness, and the weight. And we also collected the blood at different time points in order to analyze for toxicity markers or pro-inflammatory cytokines. But we are still working on that with the help of Duško. So for the moment, I only have these results, which is just the body weight, just for you to see that all the animals were growing similarly, which it is a good sign, because if they wouldn't, in that case, that would be a sign of toxicity. We did another sacrifice 1.5 months, more or less, after the inoculation to check if we could detect already the beta-catenin protein derived from the vector that we had inoculated. So we sacrificed a couple of mice, and as you can see on the right-hand side, we could detect already beta-catenin in the brain at the level of messenger RNA. This is 43 days post-inoculation, so we could already see that the beta-catenin is still there. So this is a good sign as well. We sacrificed the animals like 2 months ago or so. We took as many organs as we could, and we need still to analyze all of that. But when we were doing the necropsies, we also checked for any sign of tumor development, and at least macroscopically, we could not detect any significant observations of toxicity. So in summary, I hope everyone is understanding, but so we designed different set of therapeutic constructs that were packaged in an AAV vector and tested in patient-derived models, and we selected the most efficient. This vector was also tested for safety in a wild-type animal, and for the moment, it looks safe. And we also observed that there is expression of beta-catenin at least 1.5 months after the administration. And now we want to test this vector in more patient-derived brain organoids. We are also working on a further analysis for toxicity before going into the clinic. Thank you all for your attention. I'd like to thank, of course, the lab, because they all have helped me a lot during this journey, and also, of course, the CTNNB1 Foundation, because this wouldn't have been possible without them. The Little Miracles for Kids as well, the National Institute of Chemistry, and everyone. Thank you.